Okay, so here we're going to take a look at three different factors that could stop or slow down cell division. And one of the things we're going to see is that these three factors do not normally affect cancer cells. They only affect healthy cells. So the first one to look at is anchorage, which is when the cell is attached to a surface. So for example, here, if we put some healthy cells into this tray, you can see that the cells will not replicate and go through mitosis unless they are anchored or connected to the tray. That's the first step. The next one is called density-dependent inhibition. So density-dependent inhibition is when the cells stop dividing if they touch other cells. So you could also call this crowding. Basically, when the cells feel crowded, they're going to stop dividing so they don't grow too much. So here in this picture, again, you can see the cells, after they divide to a certain point and they cover this layer, they will stop dividing. However, if you were to remove some of those cells, they would again go through mitosis to fill up that area. But if you're looking at something like cancer cells, they actually don't stop dividing even when the layer is full. Cancer cells will ignore the signals of anchorage and density-dependent inhibition. And lastly is growth factor. So growth factor is a molecule that sends a message that tells the cells they should divide. And very simple, if you do not give the cells growth factor, they don't divide. If you do give them growth factor, they will divide. But cancer cells can divide even without growth factor. So how does this connect to the cell cycle? Well, remember those checkpoints that we looked at before. The checkpoints basically are a point where the cell can stop and make sure that everything is going well. So make sure that it's anchored, make sure that it's not crowded, make sure that it has growth factor, but also make sure that it has enough nutrients, or it could check for something like, is the cell the right size? Is there something wrong with the DNA? These are all things you could check at the checkpoints. So remember, we go around the cell cycle clockwise, and once we get to this checkpoint, the cell will not continue through the cycle unless everything is good at the G1 checkpoint. Just like when you come to school and you might see these little checkpoint barriers here, and maybe they won't let you into school until they check your ID or something like that. Okay, you lift this up to continue through the cell cycle, but you don't lift this up until you make sure everything's good. And there's three of these checkpoints to look at. There's the G1, the G2, and the M, and they're all checking for slightly different things. But the main idea is they're checking to make sure everything is good with the cell. And lastly, let's just look at growth factor here. So growth factor is going to basically cause something called signal transduction, which basically just means passing a message from outside of the cell to the inside of the cell. But the presence of growth factor is what lifts up this G1 checkpoint. If there's no growth factor present, then the cell will either stay in G1 until there is growth factor, or the cell could go over to the G0 permanent non-dividing stage.